My name is Elaine Vickers and this is my Cancer Basics video series. In this video, I'm going to describe some of the types of DNA mutation found in cancer cells. As in many of my other videos, I'll start here. Our cells contain chromosomes. Chromosomes are made from condensed DNA. That DNA contains genes, and the genes are the instructions for making proteins. In some cancer cells, the chromosomes are a complete mess, like you see here. There are extra copies of some chromosomes and missing copies of others. Some chromosomes have broken and the pieces have gotten swapped around. There are even extra bits of chromosomes that have been stuck to others. Scientists use a lot of terminology to describe exactly what's gone on. They use words like fusion, deletion, amplification, indels, and terms like microsatellite instability or MSI, or loss of heterozygosity, LOH. Now, I'm not going to go into all this terminology now, I'll leave that for other videos, but I will say that some kinds of mutations affect whole chromosomes or parts of chromosomes. Some only affect a few thousand or a few hundred DNA bases, and some affect only one. There are even changes to DNA that don't affect the order of the DNA bases, but instead affect the way the DNA is packaged up. I'll illustrate a few of these different types of mutation now. This diagram shows the result of a translocation, where two chromosomes have broken and then been swapped around. Occasionally, this means that two genes on two different chromosomes are now fused together, and instead of making two different proteins, the cell makes one fusion protein, such as the BCR-ABL fusion protein found in the cancer cells of people with chronic myeloid leukemia. Here we have a deletion, where a section of a chromosome is missing. An indel is the name for when there are much smaller insertions and deletions into the DNA, and point mutations are when only a single DNA base is affected. Changes to the way the DNA is packaged and stored inside the nucleus are called epigenetic alterations. There are hundreds of proteins involved in the packaging of DNA. Some make it more tightly compacted, others loosen it up, ready for making a protein. Many of the proteins that manipulate DNA are faulty in cancer cells and doing the wrong thing at the wrong time and in the wrong place. The most important mutations in cancer cells that affect their behavior the most are ones affecting the oncogenes and the tumor suppressor genes. Oncogenes are genes for making proteins that encourage cells to grow, multiply, and survive. They are generally mutated in a way that causes the protein to be overactive or overproduced. Tumor suppressor genes are genes for making proteins that limit how fast cells can multiply, or that cause them to die, or that help cells keep their DNA free of mutations. These tumor suppressor genes are generally mutated in a way that causes the protein to be defective or missing. To recap, cancer cells contain mutations in their DNA. These can be large scale, affecting whole chromosomes, or even down to ones affecting a single DNA base or anything in between. Or they might affect the way the DNA is packaged, whether it's really coiled up very tightly or much looser. Some of the most important mutations found in cancer cells are those that affect tumor suppressor genes and oncogenes. Oncogenes being genes for proteins that cause cells to grow, multiply, and survive. Tumor suppressor genes being genes that limit cell growth or help cells to die.